So now, as we heard this morning, stem cells may play a vital role in the future treatment of disease. The question is, where will we find enough stem cells to meet the potentially huge need? Our next speaker may just have the answer to that. The placenta is a source of highly potent and versatile stem cells in abundant quantities. Placental stem cells have many of the characteristics of embryonic stem cells without the ethical and moral considerations associated with embryonic stem cells because, of course, you don't have to destroy an embryo or developing fetus to obtain them. Placental stem cells have enormous power because of their versatility. They can differentiate and mature into all of the different specialized cell types of the body. And additionally, these cells are immune privileged, meaning they do not have to be tissue matched with the recipient. These cells are also highly expandable, which means they can be scaled up in regenerative medicine and the treatment for use. They can be scaled up to meet the need. So Dr. Bob Hariri is a pioneer in the use of placental cells in regenerative medicine and the treatment of many diseases, including autoimmune diseases and cancer. Please welcome Dr. Bob Hariri. So Bob has a presentation. He's promised to go through it quickly because I have questions for you, and I've got a hook.
Thank you, Max. Good morning, everybody. It's a real privilege to be here, and I want to thank our guests here our host here at the Vatican, and Dr. Smith for again organizing such a wonderful conference. Interestingly, I'm a surgeon by training and a scientist by practice. And when I first found out about stem cells, I did something which was probably unwise here. I felt I could be agnostic about cells, where you got them, and so on and so forth. But over the years, recognized that for stem cells to have a meaningful role in medicine, someone was going to have to productize them. Joanne just alluded to that. The demand for these products is going to be enormous. And so identifying the right source and the source that can allow us to augment the biology of an individual is essential. So at our company, we have considered that what we do when we deliver cells is we are augmenting the system. We can augment the immune system, we can augment the repair system, we can ultimately augment the longevity of an individual. And when you consider that over the last 20 to 25 years, there's been what I had considered a fog in cellular medicine because we really didn't understand how they would be deployed. It was sort of like flying in the blind, something I've done a number of times. Yet 2 decades of basic and applied science have better allowed us to understand the mechanism of action of these cells and has allowed us to recognize the way they actually drive biologic effects is by orchestrating the endogenous system, the host's own population of stem and progenitor cells. And that actually, to some extent, was a liberating understanding and discovery. The challenges in bringing these cells into the hands of clinicians, in my mind, falls into understanding what the best source of cells might be, what the biological attributes are that can be utilized to drive a system to either repair itself or to boost the response against a malignancy. And you'd need to identify a source that can be of the high enough quality to meet the demands of the clinical community and the expectations of the regulatory community. And you also need to have something that's highly scalable so that the economics make sense. Therapeutics that are outlandishly expensive just are not sustainable in any healthcare system. Now, what's changed in the last 20 years is that we've had a tremendous evolution in our understanding of how living cells exert their properties as medicines. There's also been the development of a number of pioneering technologies that allow us to harness the power of these cells. For example, you heard during this conference about immunotherapy using CAR T cells. There's also been 2 unprecedented decades of what I call intrinsic safety being demonstrated for the use of cells as medicines. And that has led to an increasingly favorable regulatory community where they recognize that, that the risk-benefit ratio when you've got a very safe product allows you to be more permissive in the application of cells in different indications. And there's an increasing adoption by the clinical community. Remember that the physicians who are in the midst of the depth of their practices today have grown up in a clinical world where stem cells and cellular medicine is well understood. So timing is everything, as it is in life. Now, my interest in placenta was launched by what I call an off-chance observation when my wife was pregnant with our first child, who's actually in the audience and is a budding scientist in her own right. I recognized for the first time, although I had seen it many, many times before, that the placenta was growing and outpacing the development of the embryo and the fetus. I'm an engineer by training. I looked at this and said, if in fact what I was taught in medical school, that the placenta was a vascular interface between the maternal and fetal system, they should develop at the same pace, the same rate. The fact that the placenta was outpacing it made me think that it was a governor in the development of the fetus. And if so, why? So that led me to understand that the placenta had a unique set of properties that could be the basis for creating the products I just alluded to. The unique immunobiology of this organ, the fact that the placenta is nature's professional allograft, meant that it could be used as a one-size-fits-all for patients. You could deploy these products without having to match every product to a recipient. It's an abundant resource. In the United States alone, there are over 4 million placentas are discarded every year. The consistency of procuring material at 40 weeks gestational age, plus or minus 2 weeks, is really remarkable and gives you great opportunity to create quality systems. And the economics, and remember, this is a waste material that we turn into valuable products. So this is an ideal platform for developing therapies. Now, consider this, because at a global conference like this, we have to understand the implications of how this tool can be used not just in the industrialized world, but even in the developing world. There are 130 million births annually where the placenta basically is discarded. Less than a fraction of a percent of placentas are utilized for any purpose. And in our hands, one placenta can produce thousands, if not hundreds of thousands, of different medical treatments. The placenta has been historically considered a remarkable resource, even in wound healing. And so you may know that the placental tissues and the membranes of the placenta have been used for decades to encourage, support, and promote healing of various tissues. Our veterinary colleagues have been using the placental membranes to heal wounds in, for example, the equine species. In our hands, over the last 15 years, we have developed a number of products from the placenta, clinical candidates, living cells, that we've used to treat a range of disorders, from autoimmune diseases like Crohn's disease, multiple sclerosis, rheumatoid arthritis, and we've advanced that to use products to treat degenerative diseases, including degenerative vascular disease. We've treated diabetic foot ulcer, diabetic peripheral neuropathy. We've also used these cells to enhance and augment the effects of a bone marrow transplant. But most recently, and in sync with what you've been hearing at this conference, we recognize that the placenta is also a source of immune cells which can be used to treat cancer, which can be used to even potentially enhance how we treat infectious diseases. And I want to speak a little bit about what we're doing in a unique product from the placenta, a natural killer cell. Now, one of the things we recognized years ago was that although there are millions of pregnancies a year in the United States, there is a small percentage of pregnant women who have an underlying cancer. In some cases, it's breast cancer, skin cancer. In some cases, they're blood cancers like lymphomas. But the rate of transmission of cancer between mother and fetus is essentially unreported. Now, in the convention, you know that most cancers are very, very good at metastasizing. It intrigued us that the placenta seemed to be a great barrier to that transfer of material. And we began to understand that part of that might reside in the unique immune cells of the organ. So we developed a product, PNK007, which is essentially a placental type of natural killer cell, which we can mass expand and use to enhance the treatment of different malignancies. Natural killer cells from cord blood and placenta have inherent benefits from natural killer cells derived from adults. They can enhance the functionality of a defective immune system. They're very easy to manufacture and produce. They're high viability, and they have demonstrated toxicity against a variety of tumors. Today, our product produced under regulatory compliant processes has shown enormous safety and quite high activity against, for example, multiple myeloma. The way these natural killer cells do it, as is illustrated in this little movie, is the little squiggly cells that are attacking the round cells and literally destroying them do so by getting up close and personal and creating a very targeted, direct killing of the cells by injecting, if you will, a perforating molecule that causes these cancer cells to die in a very, very high rate. This, we think, is one of the great opportunities to take placental source materials into the immunotherapeutic regime. We've recently had approval to start treating patients with a CD38 CAR T product, and along with my partners at Sorrento, we recognize that we can not only use the convention, which is to put these CAR constructs into the patient's own T cells, but to take it one step further and to put them on placental T and natural killer cells, which means it would be an off-the-shelf one-size-fits-all allogeneic product. That, we believe, is a revolution in immunotherapy of cancer. Now, I've often said the way we can look at what a stem cell really is that it serves as kind of a biological master boot disk. In previous presentations here at the Vatican, I've described that every cell operates and behaves almost like a small computer, with the biological software residing within the nucleus of the cell. That biological software is encoded in the DNA, and it is, in essence, processed in the cytoplasmic processor that operates like the CPU of a computer. And interestingly enough, the cell membrane acts like a keyboard, and that keyboard actually interacts with the environment, accepts information, and uses that information to drive the expression of genes, the reading of that biological software, and ultimately the production of the various molecules from that DNA. We see this as potentially serving as a programmable synthetic device, which will allow us to confer many biological properties to patients who need that. And what this means for us is that if you consider that stem cells might be at the heart of the aging process, we believe that the stem cells from placenta, because of their source being a newborn, might be the perfect way to reboot the software of an aging individual. Now, years ago, I recognized with some of my colleagues that if you look at the bone marrow of patients over time, there is an exponential decline in the number of stem cells capable of producing colonies. Now, that's a hallmark of stemness, if you will. Now, when we made this original observation, we took it a step further and actually evaluated the stem cells from patients who were being evaluated for cancer and found that this correlation was even more powerful. So there is an age-related decline in the quantity of stem cells that reside in the various compartments of our body. We took this to the next level and decided to try a little experiment, which meant that if your stem cells are declining with age, what if you boosted them back up? What would that do to your system? And this little experiment, I call it a skunkworks experiment, where we took rat placental stem cells, collected them at birth, and gave them back to the animals as they age, had a startling result. We saw that we could increase the lifespan of these animals as much as 30 to 40% with just monthly injection of their own cells, literally given intramuscularly. We decided to go and confirm these observations with one of our colleagues out at the Burnham Institute in San Diego, Dr. Evan Snyder, and found that, lo and behold, stem cells decline in quality and quantity in virtually every compartment of the body. And what's illustrated here is that muscle stem cells, brain stem cells, and bone stem cells decline significantly as a function of aging. And in muscle in particular, those measurements of endurance and strength tell you that functionality is lost with the decline in the number of stem cells. This was something that our plastic surgery colleagues recognized, because if you look at the skin of an aging individual, the functional changes, the wrinkling, the loss of the ability to retain water, that's all related to a loss of the cells that can produce the vast repertoire of proteins that are resident in that tissue. And that made us believe that there was an opportunity to turn back on some of the genes that get turned off as a function of aging by simply delivering new stem cells to these aging individuals. Yamanaka won the Nobel Prize for recognizing that as stem cells differentiate and mature, they literally go through a series of gene silencing events. Those gene silencing events create specialized cells that are very good at what they do, but not very good at doing what their neighbor cells do. So maybe you can turn that back on by delivering some of these cells collected from the placenta. So cell differentiation, that process of going from an undifferentiated primordial stem cell to a specialized cell, it increases synthetic efficiency, results from gene silencing, but results in a loss of versatility. And that loss of versatility is manifested in a restricted proteome. In other words, the number of proteins these cells make is dramatically reduced. So the physioanatomic changes that occur during aging seem to reflect the continuous renovation of tissues driven by an ever-declining population of stem cells. And if this was the case, then the reduction in stem cells might be countered by delivering exogenous stem cells, ideally from a young source. So we believe that stem cell differentiation echoes the process of aging, and that we have at our disposal a vast population of cells that can be produced from placentas, that can be delivered without having to match from donor to recipient, and can change the ratio of undifferentiated cells and differentiated cells and make the system think it's younger. So if you consider that aging is a stem cell problem, it results in a shift of that balance between the versatile cells and the specialized cells, is necessary to drive renovation of organs and tissues, and is necessary to orchestrate the response to injury, then it seems very logical that we can recharge the regenerative engine by replenishing the reservoir of stem cells that reside in our tissues but is lost with age, and that we can replace those deficient protein products that restore organ and tissue functionality. Thank you very much, and be happy to answer any questions.
So Bob, you've covered just a huge amount of territory there, and let me make sure that I'm straight and that people in the audience are straight. The cells that you derive now from placentas, there are different populations there. You have like the NK, the natural killer cells, the immune cells. There are also stem cells. What else is in that gamish?
So what I was hoping to get across was that the placenta is the best place to find large quantities of high-quality stem cells. But in addition, the organ is also a tremendous source of immune cells. And since cellular medicine, the field that we're all here to talk about, has evolved into more than regenerative medicine, but also into immunotherapy, it's nice that this same source material that's procured under very rigorous controls can provide tools for both.
So you mentioned that the natural killer cells were in sort of the CAR T cell versions off the shelf perhaps, might be effective against some cancers. Those look like, from the ones that you had on the slide, were liquid cancers, blood cancers, right?
Right.
The hard part, even in CAR T cell therapies, have been to use those against solid tumors, which are the really big ones. That's a challenge.
But you know, the interesting thing is the immune system, we know, plays the key role in defending us against malignancies, right? No doubt about that. The reason that liquid tumors are the first to be approached using this novel technology is because they're accessible, right? The cells can get to the tumors.
Exactly.
We are going to figure out ways to deliver engineered immune cells and even native immune cells like the placental NKs that they can get to solid tumors. There may be local delivery systems. There may be ways to enhance trafficking of these cells so they make it to the solid tumor. I'm convinced that it's just a matter of time before these validated, proven methods to treat cancer migrate into and become a tool for treating solid tumors.
The part that I think most of us are going to be interested in is anti-aging.
That's why I stuck it in there.
Yeah, exactly.
It's important to me, as a guy who's turning 60, it's important to me. It's striking to me that if you actually go and measure stem cells, the quantity of stem cells in every tissue compartment as a function of age, that there's an exponential decline. But it's also striking that the stem cells in those compartments as you age are damaged. They are corrupted by life on this planet. DNA in these stem cells is corrupted by viruses, by radiation, by chemicals we're exposed to. And those subtle changes to the DNA means that the quality of the products produced by that DNA are qualitatively defective. So, the nice thing about stem cells collected from birth, is once you isolate them, expand them, and put them into cryopreservation, suspended animation, they essentially are as good 20, 30, 40 years from now as they were at the time of birth. Hal Broxmeyer, one of the pioneers in cell therapy, did some really wonderful studies. When he first collected and banked away stem cells, he decided to, every year, thaw some of those stem cells and compare them to the original cells. And Hal showed that qualitatively these cells were as good 20, I think he's out now to 28 years. They're as good 28 years as they were at birth.
Unbelievable. Well, we have lots and lots of uses for our placental cells, I believe. I'll be getting my infusion of placental cells to get rid of this
You're on the
gray
list.
hair.
You're on the
I'm
list.
Good. That's all right. Dr. Bob Hariri, thank you. A pleasure.
Thanks.
Thank you.